titers have to be really high. If you get exposed to a respiratory pathogen, the titers again have to be high to protect you. For the mucosal vaccine with IgA, it's dimeric or polymeric, and you have all this low affinity interaction. And I don't think that the functional ability, you have, from an IgG response, you have to block that interaction between virus and cell. I think IgA can be better cross-protected because it can turn this one low affinity interaction to something that's high affinity because it has all these different binding sites, and two, because it doesn't have to necessarily just block the interaction between virus and cell to be functional. It can basically exclude this from the epithelial surface. It can drag stuff through the mucosa. I think that there's a lot of other ways that IgA, can turn this low affinity action of one to something high and actually be functional beyond what an IgG response. That's what I think based on our flu work.
So from NIH, it's a wonderful idea to test. So my question is, as you mentioned, when you approach a mucosal protection, the dynamic will be very different. There's a naive population versus some population has been infected with similar viruses.
Yeah.
So if we imagine like a COVID global pandemic, what is your thoughts if you are using this tablet in United States versus, for example, in like Africa or China, anywhere else?
Yeah, I mean, I think that the technology was designed with the idea that you could have a room temperature delivery service to make not just a mucosal response, but a systemic as well as a mucosal response. And we think, of course, obviously it'd be great from the standpoint of getting people to take more vaccines. If you could ship them to them. I mean, we were joking, we could send it by Uber Eats or something, the other day, it would work. But, the key thing, I think, from the standpoint of you're talking about, people have been infected over and over again with coronaviruses. And I think that's probably one of the things that creates a barrier potentially for putting stuff in the nose, because you've got all those antibodies up here, and if it's gonna bind to your antigen of choice before it hits those naive or memory B cells, it's gonna affect it. Now, we think that you can bypass the upper respiratory tract problem by going with tablets. CanSino has found that they can bypass it to some degree by using aerosolized adenovirus as well. And so we think there may be a way to bypass, but I will admit that there is the potential that your memory pool is pretty chock full and it's going to be difficult.
Yeah, thank you for that. The reason I'm asking, for example, in the cholera phase 3 trial, it was very successful in developed country, but when you put it in like Indonesia, developing country, it failed large scale trial because the mucosal bacteria.
Which one did you mention?
Cholera vaccine. It's not virus.
Oh, okay.
Bacteria. Thank you.
Oh, cholera, yes. No, there was a big, and again, I misunderstood you. Yeah. One of the things that we had thought about, of course, is that the rotavirus vaccine as well works really well, in Europe and the US. But in other countries, especially in small kids with intestinal biology that's maybe a little infected, it doesn't develop as well. The idea was you need all that inflammation is suppressing the replication, and you're not getting enough antigen. So we think because we're using a non-replicating approach, there may be some. We think that we'd be able to get through that and be able to deliver the thing. And because we don't have to replicate, we might have a better chance. Plus, there's not as much mucosal layer as well in those kids, so it might be easier. Having said that, we haven't tried it yet. We're planning to do that in the next few years.
Nice talk, Sean. What is the dose level? This is a non-replicating virus. What's the dose level per tablet?
So we're using somewhere between 10 to the 9th to 10 to the 11th, depending on the indication. So it's a decent dose. So yes.
Thanks, Sean.
Thanks.
Question, do you have any idea how you're going to be evaluating the efficacy of this vaccine compared to the other vaccine that's available there, number one? Number two, if somebody has received the injectable vaccine and you give him your vaccine, are you conferring mucosal immunity to that person, or there's some interference between the two?
Yeah, I mean, I think for approval, I don't know if we necessarily need to go head to head, from the standpoint of approval. I mean, our idea right now is to prove that it's efficacious by using a human challenge study to move it forward. If you are saying, what's happening to people that have taken mRNA vaccines, what's that doing to the memory population? I think that one of the things we saw is we could boost the seroresponses in the study. I think we did get a mucosal response, but I think it may be that we have to hit it a little harder to get that mucosal because the memory pool may have shifted somewhere. I mean, obviously one of the things we noticed is that when we induce a mucosal IgA response against SARS-CoV-2, it goes up against all the coronaviruses. So whatever is there is a memory cross-reactive response
So
in the memory pool.
is it possible to go after prevention of infection rather than prevention of the disease?
Well, that would be the ideal situation, is you could prevent infection. But the question is, okay, I know you've done a little bit of mucosal, immunity, right? Immune responses or measured them. We don't think you need a very strong IgA response to get that protection, but inducing it may be a little tricky for coronaviruses because of this history that people have been hit with all these different ones. So that's the thinking now. A little bit may go a long way.
Maybe not necessary because you're obviously superior to injected vaccine. And it's a mucosal vaccine, but have you thought about mucosal targeting? Kind of crazy concept like the recombinant fiber approach, like a peptide specific to M cells or something like that.
For targeting
with entire capsules, most of the vaccine just passes through because of that mucin layer.
Yeah.
And so that's where sort of M cell targeting becomes attractive.
No, I mean, people have basically tried, the M cell approach. In fact, with they basically have made the sigma protein and put it as part of the fiber and stuff like that. And the data has, let's just say that no one's been able to show that actually that has done anything. So it may not be that targeting M cell is basically the most important part of the process. Although, I mean, obviously we can't go inspect every human and cut them open to go figure out how, my CMO won't let me do that, so, but, that would be ideal, is basically, could we figure out essentially, what is critical from the standpoint of how to get there and how to make it more efficient. One of the things we're doing, because it's simple, is we're doing a lot of experiments now to try to basically boost the immunogenicity, because we definitely have found changing the structure or the DNA sequence of the antigen can make a huge difference for something like SARS-CoV-2. That's another way we basically dock the dose. So.
Laura Solforosi from Janssen. I wonder where your IgA come from. Where are the cells? So you generate resident cells that produce IgA in the nose, or they're coming from?
So my guess is that where the antigen is being exposed is probably the mesenteric lymph node, or basically the Peyer's patches, and then those B cells, the memory cells, or the effector cells, are going through some sort of mucosal circulation in humans for reasons that doesn't seem to happen in mice, and then those B cells get to the to nasal mucosa, and then secrete the IgA. That's what we think is happening. I mean, I can't prove let's put it this way. I know the vaccine's not getting in the nose, so we've done those experiments. So it's got to be that you're getting your activation here, and the B cells are probably homing. And certainly in the BARDA study, I can tell you, when we looked at the B cells after vaccination, 7 days, they have markers of mucosal homing. Of course, people don't know what the nasal ones are, but alpha-4 beta-7 pops up, LFA1 drops, when you get other things as well. So we think that the B cells basically in humans or in mucosa there's some sharing of that. That's what we think.
Thanks very much, Sean. Excellent talk and great discussion. Thank you to the audience.